happening, but there is some evidence for it. And it kind of makes sense. So because each of these regions of a segment, each of these regions are important for the normal body plan of the adult embryo. Okay, so any questions on what we talked about the last time? Okay, so these were the segmentation or segment polarity genes. So now we're on the selector genes. So now we're moving into the life cycle where the embryo is now becoming a larva stage. It has hatched, and most of the larvae that we'll be looking at are at this third instar stage. Okay, so just to recap where we've been, so we started out with an egg, and then we have an early embryo that's undergone gastrulation, so it has already defined body parts, and then these embryos change their shape and their form into the larva stage, and at the larval stage, we can divide the embryo into the head region, or near the acron. There's 3 thoracic segments. Okay, there's 8 abdominal segments, and then there's the telson region and the tail. So what we're going to see is how changes in the the selector genes really specify the identity of each of these segments, and how changing the expression of those genes can trick the embryo to repeat one of these segments, or to make it think that it's in a different location. And we get some really funky looking embryos. Okay, so I'm sorry, you don't see it here because it's cut off, but at this stage is where you start to see the development of what are called imaginal discs. And so these are, imaginal discs are just they're basically the onlogen of adult structures. So they are the primordial stages of adult structures, and they are located in very specific places. So you can see by the mouth parts, there are imaginal discs for the salivary glands. There's imaginal discs for the antenna and the eyes, the legs. We'll be talking about the legs and these imaginal discs towards the end of the semester when we talk about limb formation. And then there's also imaginal discs for the genitalia. And so using fate mapping, fate mapping is what allows us to know what these discs develop into in the adult fly. A haltere is a balancing organ, so it helps when the fly tries to fly. And we'll see in a few minutes that it's in a different segment than the wings, and there is a mutation that converts that section to a section that expresses wings. So you have a fly that doesn't have any halteres but has 2 sets of wings.
Can they fly without the halteres?
I don't know.
I don't like run into stuff.
I don't know if they can get off the, if they can actually, I don't know. They can actually. That good, huh? Is that on your video?
It is.
Yeah. Oh, that's going to make it the #1 YouTube.
Let's hope so.
Burp during lecture. We'll know it's you. Okay. We digress. Okay, so anyway, so the genes that control segment identity are called selector genes, and they're also called homeotic selector genes. and the family of these genes are called Hox genes. Okay, so homeotic is a term that just means the conversion of one segment to another. And homeotic genes are required throughout development to maintain specific patterns of gene expression. So these are examples of adult flies that have undergone homeotic transformation. So in this fly, which is called the Antennapedia mutant, okay, you see its head, and usually there are antennae that come out of this section. But because its homeotic or Hox genes are misexpressed, it develops legs where the antennae are supposed to be. So it basically has two legs coming out of its head. Okay. And this is the Bithorax mutant. This is the fly I was telling you about that usually, so Drosophila are two wings, they're dipterians. And in this Bithorax mutant, they have four wings because the segment that, basically the segment, I think it's T2, has been duplicated. And so it's converted one of these thoracic segments, the one that does not express a wing, to one that does. So it's a homeotic transformation. Okay, so the Hox genes are really an interesting set of genes because for several reasons. So they were first described and discovered in terms of their function in the Drosophila, but now we know that they are expressed in, and they're also expressed in specific locations in vertebrates, so their functions are conserved. But we're also discovering more functions of the Hox genes in other in specific cell types. But what they are, as you expect, like everything, they're transcription factors. They're called Hox genes because they have a specific binding motif called the homeobox. It's just a binding motif that's common to this set of transcription factors. And there's several sets of them. And so what's interesting about the genes is that, or the way that they are laid out on the chromosome, is that they are expressed exactly in the order on the chromosome as you find them in the body. So the anterior portions of the fly express the Antennapedia complex towards the anterior, and the ultratype or the Bithorax complex is expressed more towards the posterior. And nobody really understands why this is, but this order, having the physical order of the genes on the chromosome match the spatial location, the body, is called collinearity. Okay, so you can see that there's Antennapedia complex genes, so there's several, so there's 5 that are expressed, that are part of this family, and there are 3 that we'll talk about here. Okay, so they're a set of genes. And we'll talk about the Bithorax complex the most. Okay, so this is a diagram from the Gilbert text that shows you this collinearity and how the genes are expressed in the body of the embryo. So here is the genome, and the genes of the Hox genes are shown in rainbow colors, and you can see where they are expressed in the segments of the embryo and how these correspond to the different parts of the adult fly. Okay, so it's pretty much in line with their location in the body, which is just one interesting feature of this gene complex. Not all gene complexes do this, so that's why it's so unique and nobody really understands it. You know, it's like one of those head scratchers, like, well, why this complex and not all the rest? Just would make it easier for us, right? Oh well. Okay, so how do they work? So in the wild type embryo, if we look at the Bithorax genes, there's 3 sets. They're expressed in different concentrations along the body axis. So abdominal